So I'm Kerry Cameron. I'm with Southern Cross University, and my research is focusing on larval ecology of corals, specifically to figure out the best way to release coral larvae onto reefs to restore damaged reefs. And I'm looking at different aspects of larval ecology that will help us refine the method. So specifically, I've been looking at density of larvae. So if we have a tent or some kind of method of containing these larvae over the reef, what's the best density? Like, if we really crowd them, will they grow, or will they have more or less settlement, or do we just waste a whole bunch of larvae? So to look at larval density, obviously if you're going to put out great big tents that are 5 by 5 metres, you don't have a lot of that. That's pretty thirsty for larvae. So what I've been doing is shrinking it right down, and I'm doing my density experiments in actually lunchbox containers there. So we chop the sides off, we glued plankton mesh around each of the 4 sides, and then out on the reef we put a post, lunchbox, settlement tile, and then a lid on top, and then we inject different numbers of larvae into each lunchbox. So I'm going to test in the lowest density, maybe 10 or 100 larvae, and then in the really full lunchbox containers, I'm going to put in up to 5,000. And there'll be a range, so it's going to be a regression design testing that range of larvae at a small scale. So by looking at that, I'll be able to see in the volume of water, but specifically how much space or how much real estate is available in the tile. If I put in 100 or 100 settle, if I put in 5,000, obviously there's not going to be enough room. So what is that sweet spot? What's that magic number that we can maximise our settlement without wasting larvae? So I've done it once in the Philippines, and I think the magic number is between 1,000 and 2,000 with the Cropper tenuis, which is the same species as this little coral right here. And I'm hoping to try it again here in the Northern Great Barrier Reef to see if there's a difference there. So what's that number, and also how that affects the corals over time. So we know that in my experiment, putting in between 1,000 and 2,000 per container was really great for settlement, and also seemed to affect their survival over time. So the corals that had the most corals at the end, the ones that started with between 1,000 and 2,000. Most of those do die. The mortality of corals is really high in the first 6 months. But there's no way you'd want 1,000 corals on one piece of area that big. You only really want 1 or 2, but they do have a very high mortality in the early stages, and that's common. But yeah, we just want to find that magic number for maximising settlement, which then leads to maximum survival after 12 months. So this will be really handy because we know this is a really great method. We call it larval restoration. We know it's a great method, but raising larvae is hard work. It takes a week. It's lots of heavy lifting and lots of being careful with our donor corals. So we don't want to waste any of them. So the whole point of doing this is that we love coral reefs and we want to help them. Around the world, coral reefs are in a pretty bad state. Lots of them have been degraded. So there's lots of people working on coral restoration. We think this is one method. There's many methods. This is our method that we're trying to develop. So we hope that we can use this to help other people or teach other people how to use this method. So we've done it in the Philippines with great success. We've been trialling it at a very small scale in the Great Barrier Reef, Kerry Island and One True Island for the last 2 years, and now we're moving up to the Northern Great Barrier Reef for the first time this year. So one of the ways we get larvae, there's a few different ways. We can collect them in the field by going out on spawning nights and taking a spawning cone, which is a thing that you need to know which corals are going to spawn. You drop the spawning cone over the top of them, and then the spawn will float up into the cone and be collected in the jar at the top. That's a really great method if you know where the corals are and what time they're going to spawn. Another method that we have at small scales is we go and collect the gravid colonies. So the week before the corals are due to spawn, and we know that for which species fairly closely, you can go and snap off a tiny tip, and if you can see these bright pink eggs present in the tissues, you know that coral's going to spawn in the next few days. So we collect those, very carefully collect those corals and take them back to a hatchery or a lab or an aquarium, wherever it is that we're working. And then we just look after them and keep them comfortable. Then on spawning nights, we put them in isolation or nally bins or some kind of plastic container that is with still water. When they start to spawn, we just let them spawn. They spawn in these tanks. The water can't escape, so we just let them do their thing, and all these beautiful little spawning bundles, these bundles of sperm and eggs rise to the surface because the eggs are full of fat, so they're floaty. They rise to the top, and then once the corals have finished spawning, then we come in with our cups, and we sink the cups into the water, and then the sperm and the egg bundles just kind of tumble into the cups, and we take them away to the next step, which is fertilisation. So we get sperm and egg bundles from many different colonies, preferably 10 to 20 is good. After an hour, when we can test the fertilisation by looking at it. We can take really close pictures with our full cameras, or you can look at them under the microscope. When we think fertilisation is finished, then we take them out of that contained space and put them into a larger area, and that's where they can develop. And it's really cool to watch them develop. They go from just this one single cell, and that will divide in 2 to make 2, and then those divide in 2 to make 4. I can't do that with my hands. But you end up, you can actually watch the cell division taking place, and it's really incredible to see. So once we've got the larvae, they've gone through those initial cell division stages, and we put them into, I guess, a rearing rearing container of some description. So we've got ones here on land, we've got some out in the ocean that have worked really well. So we're developing that as well, just developing different techniques for rearing millions of larvae. And then they take about sort of 4 to 5 days to mature from being just a very simple little sperm egg kind of, early life stage, right through to a swimming larvae. And they start to swim in about 2 days, and they develop these lines of cilia. Those larvae are kind of football shaped, I guess, like a little slug. And they have these lines of cilia around their body, and they can actually move. They kind of rotate as they swim like little, spaceships. And they have some very limited forward motion as well. So they can actually choose when they settle to a point. They can't swim, across the pool. But once they are mature, they'll gradually make their way down through the water column. When they get to the bottom at about sort of plus 5 days, they'll start testing that bottom. And you can actually see them in lab situations. They'll just kind of swim around and kind of test the bottom. And, does that feel good? Yeah, maybe not. And they'll swim around and test it again. And they're looking for a perfect spot to settle. So other people in our group are looking at different settlement services and things we can do to encourage them to settle where we want them to settle. So that's interesting as well. When they settle, they find their good spot, they'll stick their bottom down first, and then they'll begin to metamorphose into a coral polyp. And once they're down and they start to metamorphose, they're stuck, and that's where that coral will live for the rest of its life. And then after that, the sexual reproduction stage, and from there, they will bud, and that's asexual reproduction, and that's how a coral colony will grow, by asexual budding for the rest of its life. And when they hit about 3 or 4 years old, they'll start producing their own spermic bundles and start singing. So one of the big things that I find people are surprised about when they hear about what we're doing is that corals don't actually spawn all on the same night. There's this kind of myth out there that there's this great big orgasm on the reef and everything, climaxes at the same time. Not actually true. Each species of coral is specific, and a spawning season is generally 3 or 4 lunar periods over summer, depending on where you are in the world. And some species of coral don't even all go in the same month. So this particular coral here, Acropora tenuis, I don't know it as well here in the Great Barrier Reef, but in the Philippines, Acropora tenuis has a 3 to 4 month spawning window. So some colonies will go in March, some go in April, some go in May. So yes, it's synchronised, and yes, it's mass spawning, but it's not simultaneous at the same night everywhere around the world. It is quite regionally specific. So you do have to know, when you're doing science or trying to research coral larvae, you need to have local spawning records, and you need to have good local knowledge. So there's so many things I love about science. I love finding out new things. I love travelling to cool places. I love diving. All of those things are wonderful. But it sounds super nerdy to say, but the thing that really gets me passionate about it is I love coral reefs, and they're such an important ecosystem for their own intrinsic value, but they're also really important for humanity. They provide coastal protection. They are a really important carbon sink. They provide habitat for fisheries. So many people around the world are dependent on coral reefs, and it is awful that many of them are suffering so much. And climate change is the biggest threat. There are many other pressures on reefs, but climate change is huge. So what we're doing here isn't going to fix the problem for coral reefs, but we are trying to buy them time, and we're trying to find ways to help them be more resilient, so that when the rest of the world, or the whole world, gets its act together on climate change, then we'll have tools ready to go that can help get reefs healthy again, or to help them persist in areas where they're under particular pressure. So it's just cool to be a part of that.